Okay, this chapter here is 17C. It's from this book, The Medical Reformation. This is 17C. We already did 17A and B. What we're going to mostly talk about today is more detailed discussion of excitotoxins, and we're also going to talk about my theory, the Peter Rogers, MD theory of dementia. What causes neurodegeneration? Okay, first a quick review of calcium metabolism inside a neuron. You need to know this. This is important because everything kind of revolves around calcium metabolism inside the neuron. We spoke earlier about the minimum wage ions are potassium and sodium and to some extent magnesium. They sort of are the laborers, the manual laborers doing all the dirty work to keep things functional for the neuron. And then calcium is like the aristocrat. It comes in and it basically turns the neuron on or off. Okay? So a typical sequence of events here is glutamate will come from another neuron to bind the receptor. It'll first bind what is called an AMPA receptor. An AMPA receptor is a sodium channel, and it'll allow sodium to come into the cell. When the sodium comes into the neuron, it will depolarize it, meaning instead of being negative 65 millivolts across the membrane, the charge, it'll move up to, let's say, negative 40 and keep going up. As it becomes more positive, the charge inside the cell, the magnesium that was sitting in the center of the NMDA receptor will pop out because it was attracted to that negative 65 millivolt charge inside the neuron. Once that's gone, it just bounces out of the receptor, loses its intense attraction to the center of the neuron. So once the magnesium is out of the cell and you've simultaneously got a glutamate neurotransmitter bound to the NMDA receptor, then it'll allow calcium to come in. By the way, glycine also binds to the NMDA receptor, and that's thought to be part of the mechanism why GP, as in glyphosate, glycine phosphate, the herbicide sprayed on soy and other non-organic crops. It's even sprayed on non-organic oatmeal, sprayed on non-organic beans. That's why I recommend organic oatmeal only and organic beans. It is thought to have an excitotoxic effect. Excitotoxicity refers to anything that increases cytoplasm calcium. Typically things that increase cytoplasm calcium entry into the neuron. However, in my opinion, it is wise to think of anything that prevents you from lowering your cytoplasm calcium as also being essentially like an excitotoxin. I'll explain what that means in just a moment, but basically the NMDA receptor is the big entry site for calcium into the neuron. And this happens normally under appropriate circumstances, and that's good. That's how a neuron's supposed to function. But you can see where problems will start to arise if you're getting more of this glyphosate stuff activating the NMDA receptor. That'll cause more calcium to come into the cell. If you have an abnormally increased amount of glutamate activating the NMDA receptor, you're going to get more calcium coming into that cell. You can have increased glutamate because of MSG in food, because of MFG. MSG is monosodium glutamate. MFG is manufactured free glutamate, which just means more freed up glutamate because you processed a high glutamate protein, broke it up into little bits so its glutamates are now free. And some of them do get across the blood-brain barrier or areas of the brain that don't have blood-brain barriers, so you can get increased activation of the NMDA receptor in this way. Aspartame, the sweetener, activates the NMDA receptor. Okay, so there's additional things activating this NMDA receptor, and then we're going to talk about things like here's the endoplasmic reticulum in a neuron, and the endoplasmic reticulum will have pumps to pump calcium away from the cytoplasm. Well, if you inhibit those pumps, they're called circuit pumps, sarcoplasm, endoplasmic reticulum, calcium ATPases, it's going to be harder to get calcium out of the cytoplasm. There's also plasma membrane pumps. I'll show these in additional pictures later, but there's pumps to pump calcium out of the cell. There's also something called a knockout exchanger, which we showed earlier in this chapter. And so if those are inhibited, then you're going to have a hard time pumping calcium out of the cell. Also, you need to make tons of energy because you're always pumping calcium against a gradient in order to get calcium out of that cytoplasm. So if your mitochondria are inhibited, as they routinely are by over 50 different things that are many of them relatively common in the modern world, you're going to have a hard time pumping out your calcium, and that's going to have an excitotoxin effect. Okay, I'll explain this all in more detail, but just remember, it all revolves around calcium, because the calcium is activating that neuron, and when it's done for normal, appropriate reasons, that's good. But when it's overactivated because of drug effects, toxin effects, EMF effects, and the mitochondria is inhibited, you don't have the energy to pump it out, you can overactivate that neuron. And when the neuron's overactivated, if that goes on a prolonged amount of time, the neuron will just die. It'll go into apoptosis. That's called programmed cell death. And I believe that's the main reason that people are losing brain cells. And by the way, I'm a neuroradiologist. I've been studying this for many years. I've been interested in why do people become demented for decades. Okay. Alright, so positively charged ions are called cations. So sodium, potassium, and magnesium are all cations. Okay, here's another picture. We'll go through a little more detail about what's happening with calcium inside of neurons. So let me see if I can move this little thing a little more. Alright, so we talked about how the sodium-potassium ATP pump helps maintain the resting membrane potential in a neuron. And it has to be negative, typically around negative 65 millivolts. Okay, and it pumps in 2 potassium while pumping out 3 sodium. So 3 positive charge are pumped out for every 2 pumped in. So over time, that contributes to a negative resting membrane potential inside the neuron. Okay, and that negative resting membrane potential, negative 65 millivolts, is what attracts the magnesium to want to come into the cell, and it'll sit in the center of the NMDA receptor and block it, and that'll block calcium entry into a cell. And here's the pumps I was referring to a moment ago, the knockout exchanger, Na for sodium, Ka for calcium, and that will pump calcium outside of the cell. It utilizes the gradient made by this potassium-sodium ATP pump. In addition, you're going to have magnesium binding the phosphates in the ATP. ATP is adenosine triphosphate. And basically the phosphates have a big negative charge on them that wants to pull apart from each other. And the 2 positive charges of the magnesium hold them together. It's like holding back the reins on a powerful horse. Okay, you need the magnesium. If you're deficient in magnesium, and most Americans are deficient in magnesium, that's a problem. There's less magnesium available to hold their ATPs together. So ATP-dependent processes like this pump can be decreased in function. There's less magnesium available to sit in the center and block NMDA receptors. So you can have increased NMDA receptor activity, increase allowing of calcium to enter into neurons. That's bad. You don't want that. All right, so normally under normal circumstances, when calcium is increased in the axon terminal, these glutamate neurotransmitters, which are stored in vesicles, will travel to the plasma membrane. The vesicle merges with the plasma membrane, and that releases the glutamate into the synaptic cleft. It'll diffuse across the synaptic cleft and bind the receptors on the postsynaptic neuron. Typically, it'll bind the AMPA receptor, which is primarily a sodium channel, and that allows sodium into the neuron. It's a positive charge, and this is a negative resting membrane potential, so it makes the postsynaptic neuron go up in its charge, so become less negative. And as that happens, that's called depolarization. It was polarized, being negative. It becomes depolarized, and the charge starts drifting upward. As that happens, the magnesium pops out, and there's already glutamate and glycine bound to the NMDA receptor. Now calcium can, charge into the neuron. The calcium wants to come in because it's attracted to the negative charge there. Also because of the incredible concentration gradient. The calcium concentration in the extracellular matrix is about 15,000 times. That's not a misprint. 15,000 times higher than it is inside the cytoplasm. So ions will move towards the charge that attracts them, but they'll also try to equalize their concentration gradients. So there's both an electrical attraction by the negative 65 millivolts inside the cell, and there's also, a giant chemical gradient of concentration to have calcium come into the cell. So normally calcium come into the cell and it'll do something that it's supposed to do. But when you have excessive amounts of calcium in the cell, it can activate something called calpain. And that's a perfect name for the calpain, this enzyme. So calpain, it's activated by calcium. pain because it has painful consequences. It can do things like damage the knockout exchanger and create a vicious cycle whereby you can't adequately pump the calcium out. And as it keeps on increasing the metabolic activity of that postsynaptic neuron, it can cause it to die. Okay, another thing is that excessive psychological stress. Well, first of all, the NMDA receptor is very useful. It's actually part of our circuits, our brain circuits for learning and memory. We really need it. It does good things under normal circumstances. However, when there's abnormal circumstances, all these things start adding up together and you get overactivation of MDAR. It's often called MDAR, NMDA receptor, and the R is just for receptor. So it'll often be written as MDAR, NMDAR. People call it that. Excessive psychological stress will also cause increased cortisol, and that'll result in increased glutamate being transmitted at the synapse. Synapse just means a connection between two neurons. And so what I'm saying here is psychological stress, sleep deprivation, and caffeine all have a very similar effect. They increase cortisol and they increase catecholamines, and this leads to increased glutamate going across this synapse, so the increased activation of the postsynaptic neuron. And this is what I meant by people get nickeled and dimed to death and they don't even know what happened to themselves. Death by 1,000 cuts. And people are kind of programmed their brains to think 1 + 1 = 2. But in real life, it's more like this plus this plus this plus this plus this plus this, all exerted damage. And the good news about all that is if you simply take the time and the attention to be aware of these things, you can avoid almost all this stuff. So MSG is monosodium glutamate, and what really matters is the glutamate. That's the neurotransmitter. About 90% of brain neurotransmitters are glutamate, and it is excitatory. It increases activity of the postsynaptic neuron. MFG just means manufactured free glutamate. And where that comes from is you'll take a baseline protein like gluten, let's say. It's called gluten because it's got lots of glutamate in it, and you chop it up into pieces by processing it, and then that frees up the individual glutamate amino acids, okay? And you can do the same thing with casein, the milk protein, and get that effect. You can do the same thing with soy and get that effect. You can do the same thing with whey protein and get that effect. You can do the same thing with corn protein and get that effect. So what I'm saying is processed food tends to have lots of glutamate. I talked about this in other areas, but basically you've got taste receptors in your mouth for glutamate and even in additional parts of your intestinal tract that activate reward neurotransmitters when you taste more glutamate. And so that's because our ancestors were worried about starving to death and we're happy to find a good protein source. But in the modern world, people are overloaded with food, and the food companies have learned that by having increased glutamate freed up for these taste receptors, the people will become addicted to the food. It tastes good. It's sometimes called umami receptors as well. Okay, well anyways, the more glutamate you ingest, and especially when it comes in as a bolus, it overwhelms the intestinal tract's ability to sort of sequester it. And you can get very high levels of glutamate in your blood. And some of that can get across into your brain and help contribute to causing overactivation of your postsynaptic neurons, like in your hippocampus, your memory center, for example. Okay. All right, so we talked about calpain. Overactivation of a neuron is called excitotoxicity. All right, and another thing is that, we talked about aspartame doing that as well. Aspartate's the excitatory amino acid as well. So whenever you get increased calcium inside the cell, you'll start getting activation of that cell to do whatever it normally does. So the typical thing a neuron does is release a neurotransmitter, and increased cytoplasm calcium will do that. In the pineal gland, release more melatonin. In a mast cell, release more histamine. In a pancreas, more insulin. In a skeletal muscle or a vascular smooth muscle or a cardiac muscle, cause contraction. Okay, so it activates the cell to do whatever it does, like flipping on a light switch. Like the aristocrat ion comes in and starts giving orders and bossing around everything else to do what it's supposed to do. Okay, and then how does the cell get rid of the calcium in the cytoplasm? It can pump it into the extracellular matrix, especially with the knockout exchanger, very fast. It can pump it into the endoplasmic reticulum. It's called a circa pump. You really could call it ERCA, endoplasmic reticulum calcium ATPase. The sarcoplasm part of the word sort of is a holdover from its function inside of muscles. But if you inhibit the circa pump to get calcium into the endoplasmic reticulum, or if you inhibit knockout exchanger, you're going to have a harder time getting calcium out of the cytoplasm, and the cell can become overactivated. That's what excitotoxicity is all about, overactivation of a neuron. And then remember that this knockout exchanger was dependent on the ATP pump over here for potassium and sodium. Okay, so this is primary active transport because it uses ATP, and the knockout exchanger is called secondary active transport because it's based on the gradients produced by the potassium-sodium ATPase. All right, this is also an ATP dependent, therefore primary active transport. This is the plasma membrane calcium ATPase pump to pump calcium out of the cell, and this is the circa pumps right here, and these also require ATP. You always need ATP to pump calcium because you're always pumping it against a gradient. You know, you're pumping it here against a 15,000-fold higher gradient, and it's a very high concentration in the endoplasmic reticulum as well. And the significance of needing ATP to pump calcium, what that means is you need good function in your mitochondria to make that ATP. So anything that inhibits your mitochondria will lower your ATP production and will thus decrease your ability to get calcium out of the cytoplasm, okay? And then another thing too, people say, well, why am I making such a big deal out of this? And I'll tell you why. It's an endless amount of cognitively impaired people. Cognitive impairment is going way up in its frequency. And I can tell you, like I said, in my personal experience and my internal medicine, doctor buddies, friends, most of the people, some of them will tell you virtually every patient they have over 60 is cognitively slow. There's a lot of really mentally slow older people, okay? And that's not good. The more mentally slow, cognitively impaired a person gets, the less likely they'll ever fix their problems. Plus also, in my experience, the lay public kind of has this idea, oh, you know, you go to the doctor and they give you a medication and then you get better. No, that's actually not true. Most people never get better. Most people do not age well. Most people just crash and burn, and they progressively get worse, and they get stupider, and there's actually zero hope that they will ever get better. So that's why you want to learn all this stuff so you don't end up like that. Because it's easy to avoid most of the stuff if you have a simple understanding of it. You know, there's not a guarantee you'll avoid it, but your odds will dramatically improve. Okay, all right, so now we're gonna, let's see, this slide's a little bit more of the same. You know, I'm sort of showing the same thing from a little bit of a different angle. Just a reminder, you know, the difference in the gradients of the concentration. So here's the plasma membrane calcium ATPase pumping calcium out. Here's the circa pump pumping it into the endoplasmic reticulum. It has binding proteins in there, so it's hard to know the exact concentration. And then there's the extracellular matrix, and the knockout exchanger pumps it out there. Okay, let's see here. Okay, we talked about circa being sarcoplasm, endoplasmic reticulum, ATPase. The sarcoplasm word comes from the muscle cells. All right, some of this is a bit of a repeat, but it was a real key point. It always takes energy to pump calcium out, meaning you need good mitochondrial function. All right, so now I'm going to start showing how all this stuff is coming together. Basically, you have excess dietary calcium that's going to lead to some increase in your calcium in your cytoplasm. Not that much just from the dietary sodium. Ischemia is a much bigger deal. Ischemia means a lack of oxygen delivery to neurons. That'll happen from, number one, from, let's say, diabetes. Tons of people got diabetes. Most over 60s are pre-diabetic or diabetic in the Western countries. Number two, from hypertension. Hypertension injures the arterial wall, leads to atherosclerosis. That'll lead to tissue hypoxia, okay? High-fat meal will cause some tissue hypoxia. Obstructive sleep apnea will cause some tissue hypoxia. All of these things will have the net effect of causing increased calcium in the cytoplasm. They're going to inhibit mitochondrial function. That mitochondria can't make ATP if it doesn't have oxygen to work with, okay? We talk about a lack of dietary potassium and magnesium will lead to decreased function of the potassium sodium ATP pumps, okay? So you're going to get increased calcium in the cytoplasm. The reason I'm going through this is that just to kind of make it clear, there's a lot of things happening to increase that cytoplasmic calcium, which are going to increase that neuron's activity. And this is how people are getting nickel and dime to death. All right, so what do you got to do to fix this problem? Eat more plants, so you'll get your potassium and magnesium. That's where they come from plant foods, okay? Don't add so much sodium to your food. Don't eat processed food. Most people get about 75% of their sodium from processed food. Avoid all that stuff, okay? Avoid things that cause leaky gut and leaky gums. Those open up the blood-brain barrier. They'll allow that postprandial bolus of MSG, MFG they'll help prevent it from getting into your neurons, into your brain tissue, your brain parenchyma. Okay, it'll help prevent you from activating dormant bacteria, which contribute to damage to the blood-brain barrier. And they also contribute to causing problems with beta amyloid metabolism. And they're going to have a net effect to promote excitotoxicity. Beta amyloid protein itself, when precipitated, can have an excitotoxin effect and allow more calcium into the cell cytoplasm. Okay, excessive iron, excess Fe for iron, that will contribute to this process. Okay, excitotoxins, typical ones are MSG, MFG, aspartame, the sweetener. GP is glyphosate on the non-organic food. That's glutamate itself. Okay, aspartate, some stimulants will do that as well. We talked about stress, sleep, and caffeine, all increasing cortisol, increasing glutamate transmission across neurons in your memory center, your hippocampus, for example. SERCA inhibitors, and there's a lot of them. Anything that smells bad will often be a SERCA inhibitor. They will then decrease your ability to pump calcium out of the cytoplasm, so cytoplasm calcium goes up. Mitochondrial inhibitors, and there's over 50 of them, and many of them very common, like SSRIs, statins, a whole bunch of common things. I'm going to talk about that in the next chapter, mitochondrial inhibitors. But there's lots of them, and lots of them are very common. Excess saturated fat, for example, they're going to lead to an accumulation of calcium in the cytoplasm because you can't make enough ATP to pump it out. Traumatic brain injury will have some of these effects. It'll also cause some localized ischemia in the area of injury. Cell phones, Wi-Fi, EMF, they will open up these VGCC. That means voltage-gated calcium channel. So they allow more calcium to come into the neuron. Activation of these pathways, which can also happen from EMF, is the nitric oxide peroxynitrite pathway. It's associated also with excessive reactive oxygen species. Volatile organic chemicals can do that as well. Some of these things that smell bad are part of that whole picture. Anyways, these are all things that are going to lead to increased cytoplasm calcium. This is one of the reasons why I'm a bit of an absolutist about I want to do everything possible to be healthy. So I would never drink anything with aspartame. I would never eat anything knowingly with MSG or MFG in it. I would never knowingly eat GP. I want to avoid all these things. I don't add any sodium to my food. I eat lots of plant foods, okay, because I want to avoid all this stuff. I avoid doing anything that's associated with TBI, traumatic brain injury. I avoid all the mitochondrial inhibitors as much as I possibly can. I avoid all the SERCA inhibitors because I want to make things as good as I can. I'm 60. My brain works quite well. You know, I have a lot of young people in their 20s tell me, oh, they have brain fog, they can't concentrate. The only thing that they read is like, for example, on my video channel, a lot of the shorts, I think they're kind of stupid and vulgar, but a lot of young people tell me, oh, the only thing we look at is shorts. We just don't have the time or the attention span for the longer videos. And what I'm saying is, they're in their 20s, okay? Martin Pall is predicting a worsening progressive epidemic of cognitive impairment and dementia from all the increase in EMF people are exposed to these days. Okay, let's keep going here. Things that cause leaky gut increase intestinal permeability. They'll also cause leaky blood-brain barrier due to lack of dietary fiber. Things that go in the opposite direction cause leaky brain, due to TBI, traumatic brain injury, or stroke. They'll tend to cause leaky gut, which is a double whammy of negativity, because then you'll allow toxic things from the gut to get into the blood, and then because the blood-brain barrier is open, to get into the brain tissue, the brain parenchyma. Okay, and now here's even more stuff opening up calcium channels or causing more calcium in the brain neuron cytoplasm. Okay, I wrote all this stuff down. There's just tons of stuff. So a lot of these psychiatric drugs can make this all worse. Post-traumatic stress disorders, kind of like excess psychological stress, corticosteroid medications, beta amyloid protein, some of these organophosphate carbamate pesticides. Some of this is a repeat here. We talked about SERCA inhibitors. Okay, we're going to peroxynitrite. We're not going to get in that much right now, but being more vulnerable to oxidative stress worsens all of that. Oh, formaldehyde. Watch out for formaldehyde. That's why I'll never wear a shirt, like a dress shirt, that says no iron needed. Because when they say no ironing needed, that means they got formaldehyde in there, okay? Also, I don't think medical schools and other health related graduate schools should have cadaver labs. And it's not because of, I'm not talking about the cadavers. What I'm talking about is they put formaldehyde in the cadavers. And formaldehyde's, it's a neurotoxin, okay? So that's not good. Volatile organic compounds. A good rule of thumb is anything that smells bad is bad for you, okay? Your nose is your friend. It's telling you the truth. And if something smells bad, it's bad. Okay. Wireless is not in your best interest. It's not a big deal by itself, but it adds up. Usually it's not a big deal by itself, but it adds up. Okay, so here's the neurovascular uncoupling theory, and you'll need to see my hand gestures for this. And it's actually kind of simple. And a lot of people have done research on this. And, you know, sometimes it's been called the calcium hypothesis, excitotoxicity hypothesis, but I really took it to a much higher level than what had been previously known and available. So here's how it works. You got a baseline, okay, you got a baseline metabolic rate in your neuron. And then you normally have neurovascular coupling. What that means is the oxygen and glucose delivery, the blood flow, is equivalent to the metabolic rate of the neuron. There's coupling, and that should always be that way. All right. I labeled in the slide here GOD as glucose and oxygen delivery. And I kind of jokingly, usually it's called OGD. I jokingly made it GOD. You know, obviously that spells something else. Okay, MR is for metabolic rate. So if you've got something, here's your metabolic rate, here's your oxygen and glucose delivery. If you've got things that drop oxygen and glucose delivery, it's going to make it harder for that neuron to produce enough energy to match its metabolic rate. Also, if you do anything to inhibit the mitochondria or the circle pumps and whatnot, you're going to have an equivalent effect. Like if you inhibit the mitochondria, you can't make ATP as well. So that's going to make you less able to match this metabolic rate. And the point is, the wider this gap gets between the metabolic rate of the neuron and the oxygen and glucose delivery or ATP production, the more likely this neuron will say, cannot continue, death. You know, activation of calpain, destruction of the NOx exchangers, and the cell just dies. It'll often happen gradually, and the cell will go into apoptosis. Apoptosis means gradual programmed cell death, whereby the cell recycles its chemical components so they can be used by other cells. They're sort of absorbed, phagocytized by the microglia, the immune system, and then sent elsewhere in the body to be used by other cells. And when you have that occurring, and it happens all the time, you just see the brain shrinking and shrinking. There's really nothing to see other than a shrunken brain. If you look at a microscope, you just see fewer neurons than expected. And that's totally different than a stroke. A stroke is a sudden occlusion of a blood vessel, wiping out all the blood supply to areas of the brain, and it dies real fast. When a brain cell dies fast, it can't control its death. It becomes hyperosmolar, it swells, it bursts, it lyses its plasma membrane, all of its inner contents spill out into the extracellular matrix. That causes edema, fluid accumulation from inflammation. I can point on a brain MRI. A stroke happened right here. I can tell you what blood vessel it was, what gyrus it is on the brain. So that is sudden death of brain cells, also called necrosis. But that's not the most common thing. The most common thing I see with these dementia patients is this progressively shrinking and shrinking brain. We call an old brain a loose brain. By loose, what I mean by that is because it's shrunk, there's now a lot more fluid around it. Whereas a young brain's a tight brain. It's uptight up against the skull because there's not that much cerebral spinal fluid around it. So anyways, my point is very commonly, I see many, many thousands of demented brains, is you just have this shrunken atrophic brain. Okay, so what are the things that will drop oxygen and glucose delivery? We talked about a lot of these. You know, if you overtreat hypertension, you drop perfusion of the brain. So you don't want to overtreat that hypertension too low. I mean, why do you have blood pressure in the first place? To get blood up to the top of the brain to go against gravity. So if you give too much medication, drop the pressure too low, you don't perfuse the brain adequately, that's a drop in glucose oxygen delivery. If you let the blood pressure get too high, you'll damage the arteries in the brain, and then that response to the injury will lead to atherosclerosis, a blood clot, increased collagen in the wall of the artery, and that'll also decrease oxygen and glucose delivery. Hypertension is the most common risk factor associated with intracranial atherosclerosis in these small silent strokes. Diabetes will also damage your brain capillaries. It'll cause thickening of the capillary basement membrane. I've talked about this in other lectures, and what that'll do is that'll lead to less oxygen being able to reach the brain cell. So when you got less oxygen getting the brain cell, that means less oxygen getting the mitochondria, and that means less ATP production. And if you have less ATP production, that means you have less energy available to pump that calcium out of the cytoplasm and turn off that brain cell. Okay, just aging in general. As people get older, they tend to have a decreased functionality of their metabolic systems, especially, if they've lived unhealthy, eating a poor diet all these years, lack of exercise, lack of sleep, etc. Okay, there's some real common conditions. Atrial fibrillation means abnormal rhythm in the atrium of the heart. So you can lose like up to 25% of ventricular filling to have decreased cardiac output, less ability to get adequate blood to the brain means less ability to get adequate oxygen and glucose to the brain. Okay, a lot of AFib is due to ischemia, which means atherosclerosis in the arteries of the heart. So you want to prevent all that stuff. So basically, all the good stuff helps everything in your body. Okay, and all the bad stuff will hurt multiple things in your body too. So you want to only do the good stuff, avoid the bad stuff. Okay, CHF is congestive heart failure. It means the heart's not pumping as effectively. Let's say it's dropping its cardiac output, its stroke volume. Well, you're not going to get as much blood to the top of your brain, and that's going to decrease oxygen glucose delivery to your brain. Aortic regurgitation, aortic stenosis. So that means the aortic valve. So the heart contracts and it pumps blood into the aorta. And if you have aortic regurgitation, means the blood's leaking backwards into the heart, or stenosis, meaning it has to pump through a tight narrowing, you're going to get less blood to the brain. Okay, a drop in glucose and oxygen delivery. All right, if you have inhibitors of SERCA, remember SERCA was for the sarcoplasmic endoplasmic reticulum calcium ATPase, and that's to pump calcium from the cytoplasm into the endoplasmic reticulum of the neuron. So if that pump's not working, more calcium accumulates in the cell cytoplasm, cells overactivated. Mitochondrial inhibitors. If you're inhibiting the mitochondria, it can't make as much ATP, so you don't have that ATP available to run your potassium sodium plasma, your plasma membrane potassium sodium ATP pump, thus less energy to run your knockout exchanger. You can't get that calcium out of your cytoplasm. Cell stays overactive. You can't run your plasma membrane calcium ATPase either. Cell remains overactive. Okay, low potassium and magnesium because you don't eat enough plants. That'll make it worse as well. You can't run your potassium sodium ATP pumps too well. If you have got excess iron, you'll have more oxidative stress, which will have a tendency to damage your mitochondria. For example, through the Fenton reaction, production of hydroxyl radicals, and then damage the inner mitochondrial membrane. If you've got excessive peroxynitrite, that will also damage mitochondria. Okay, and EMF will also lead to opening of the voltage-gated calcium channels, flooding calcium into the cells, especially if it's pulsed EMF compared to continuous EMF. All right, so that was a drop in either the oxygen and glucose delivery to the neuron or a drop in ATP production. Okay, so all of those things will lead to, let's say here's your metabolic rate, there's going to be a drop in energy production, glucose and oxygen delivery. So that's bad. But it gets even worse than that because there's stimulants that are going to raise this metabolic rate, further increasing the amount of uncoupling between metabolic rate and oxygen glucose delivery hyphen energy production. So things that will increase the metabolic rate of the neuron: increase MSG, monosodium glutamate in your food; increase MFG, manufactured free glutamate in your food; excessive psychological stress, raising the same hormones, cortisol and catecholamines, and you get the equivalent effect with sleep deprivation, equivalent effect with caffeine, equivalent effect essentially for the glutamate with corticosteroid medications. Okay, all of these things are going to impair cognitive function. And you can think of a person with PTSD, they're perseverating about something that they're stressed out about. Well, they're overactivating those neurons, and that's why they'll have decreased cognitive function at that time. Tobacco is a stimulant. Amphetamines are stimulants. ADHD meds are stimulants. We talked about glyphosate causing increased activation of the NMDA receptor, letting more calcium into the cell. We talked about BAP, beta amyloid protein oligomers. Those also will increase the amount of calcium getting into the cell. And in my earlier slides, I labeled more things that are contributing to this. So all those things that increase calcium getting into the cell are going to increase metabolic rate. So you see what's happening simultaneously. Like, look at caffeine. It's a double screw job. Caffeine is going to increase the metabolic rate with its stimulant effects, but it's also a vasoconstrictor, so it's going to lower the oxygen and glucose delivery to the brain cell. It's a double screw job, okay? So you see here, once you've got the metabolic rate quite a bit higher than the glucose oxygen delivery as well as energy production ability, mitochondrial function of the neuron, that neuron just say, I can no longer function under these conditions. Calpain's activated, destroying things all over the place. The neuron dies, okay? And people lose a lot of neurons that way. And you can see that this is sort of an all-encompassing hypothesis that explains a whole bunch of stuff. And it tells you exactly what to do. Avoid the high-fat meals. Avoid the excessive dietary sodium. Eat your plant food so you get your potassium and your magnesium and your fiber. Avoid excessive iron in your food. Avoid circuit inhibitors, anything that smells bad. I'll talk about that in a separate lecture, but that's the gist of it. Avoid the mitochondria inhibitors. I'm going to talk about that in the next lecture. There's like 50 of them. Okay? It tells you what to do. Don't be a moron. Don't smoke cigarettes. Don't give attention deficit medications to your kid. Try not to have to take corticosteroid meds if you can. Maybe you need them, but if you can, you might be able to fix your problem by fixing your leaky gut. Don't drink caffeine. Avoid processed food with MSG and MFG in it. Avoid insulin resistance because that increases beta amyloid protein accumulation and precipitation, which means avoid dietary fat. Avoid glyphosate means avoid the non-organic foods that have that. You don't have to eat organic for everything, but I would certainly eat organic if you're going to eat the foods that would otherwise be sprayed with this glyphosate stuff. It also increases fatty liver. It also increases leaky gut risk. Okay, let's see here. Oh, and people say, well, what about the blood-brain barrier? Doesn't that protect you from MFG getting into the brain? Well, there's areas that don't have a blood-brain barrier, like the circumventricular organs around, especially the third ventricle, because they're chemoreceptors, so they have to be able to take up more chemicals from the blood. Area posterior of the vomiting center, the pineal gland. That's partly why the pineal gland gets destroyed relatively early by excess F- in the water and ends up being calcified. Okay, high-fat meal also can increase blood-brain barrier permeability. Diabetes will increase blood-brain barrier permeability. Hypertension will increase blood-brain barrier permeability. Young people don't have that well-formed of a blood-brain barrier, but old people got tons of reasons to have damage to their blood-brain barrier. Okay. Other stimulants we didn't even talk about, these methamphetamine, cocaine. So yeah, don't ever use those recreational drugs. They're all bad. They're all brain toxins, okay? And yeah, Craig only figured out the concept of excitotoxicity back in the 1960s. And he did a lot to try to reduce its use in processed food, but the food companies sort of had the money to keep it going, even though they at least decreased it in baby foods. And remember, there's a lot of things happening simultaneously. People are being exposed to a lot of aluminum simultaneously, which is also a mitochondria inhibitor. And it's toxic in other ways as well. F- is toxic in other ways as well, in addition to being a mitochondrial inhibitor. Okay, Blalock wrote a great book in 1997, Excitotoxins, and he has a really good YouTube lecture on excitotoxins, okay? And he emphasized especially MSG and aspartame. There's a lot of excellent PhD scientists, people like Mark Manson, Dennis Choi, Danton O'Day, who've done a lot of research on excitotoxicity. Okay, this lady, Catherine Reed, she's a PhD biochemist specializing in proteins. She's the one who figured out that MFG was the cause of autism in her daughter. Autism can be caused by different things, but it can sometimes be caused by excessive exposure to glutamates. She wrote a great book on that. I think it was Fat, Sick, and Stressed. And I've made videos about her. She has a good YouTube channel. She has a real good TED Talk video on YouTube where she goes through all of this stuff. Okay, and she actually helped me to better understand manufactured free glutamate. This other guy, Martin Pall, he's a PhD, sort of a biochemist and a physicist. Guy's a genius, and he wrote a good book all about the nitric oxide peroxynitrite pathway, which is sometimes called the no-oh-no pathway, and how that relates to neuronal oxidative stress and neurodegeneration. The bottom line is try to decrease your exposure to EMF to the extent that you can. You probably can't control it too well in the day, but try to control it at night at least. That's why I even put a Faraday cage around my bed. And I've tested it too, that I have essentially like 0 according to my tester results, EMF in my bed. Let's see, what else around here is a little bit interesting. Douglas Calatheresia Pretorius did a great job of studying prions and amyloid blood clotting, oxidative stress-related neuronal damage, and they believe that also damages the blood-brain barrier and leads to increased excitotoxicity. The enzyme that clears insulin out of the blood, insulin-degrading enzyme IDE, it also removes beta amyloid protein from the blood. So when you have insulin resistance and higher levels of insulin in the blood, IDE is more attracted, has higher affinity for insulin than it does for beta amyloid protein. So it can only be made in finite, limited quantities. So when IDE is used up to clear insulin from the blood, there's less available to clear beta amyloid protein from the blood, and some of that gets back into the brain, and that opens up mDAR channels in the plasma membrane and neurons and causes increased calcium in brain cells. So all these things are working together. This lady, Stephanie Seneff, PhD from MIT, did a lot of research. She wrote a very interesting book. I think it's a really good book called Toxic Legacy, where she goes through all the details of the different toxic aspects of glyphosate. Some people say, oh, I should call it glyphosate. I like to call it glycine phosphate because that tells you what's relevant for understanding its chemistry. This guy, Dean Murphy, did a lot of work showing the problems with F- fluoride and how that's a brain toxin and a mitochondrial toxin. I'm mentioning all these because these are all contributing to excitotoxicity. And also, this is why I think I deserve credit for having the best theory of what causes dementia, because I figured out all this stuff and connected it all together. A lot of these PhDs did fantastic research about the ion channel or something. Okay, great. You know, Blaylock did a great job of teaching everybody about excitotoxins with regard to MSG and aspartame, okay? But I'm able to see the entire picture from all the nutritional issues, the toxin issues, the neurophysiology issues, the brain MRI issues, etc., to put it into an all-encompassing theory. Brunda Brody, she's the one who had a child who was had essentially something like chronic fatigue syndrome due to SERCA inhibitors. And then once she understood that and she was able to reduce the child's exposure, her kid did really well. Her kid had to drop out of grade school, but then he went on to graduate from college with a degree in engineering. And engineering degrees are about the most intellectually difficult degree you can get in a college. So that's pretty impressive. She was a pretty wealthy, smart lady, not any work in the healthcare business, but she hired some scientists who figured out what was wrong with her kid, and she followed their advice and cured her kid. Pretty impressive. Not many people are cured from chronic fatigue syndrome, and she figured it out and cured her kid. Okay, Danton O'Day, he's another researcher who's done a lot of good work on calcium calmodulin, and we didn't even really get into that. That's the whole idea of the metabotropic receptors. Okay, there's ionotropic receptors for glutamate that open up ion channels, and there's also something called metabotropic receptors that run through a cascade of reactions. They have some similar effects, though, to increase cytoplasm calcium. Okay. Like I said, I'm the one who figured out the whole thing and put it into the neurovascular uncoupling hypothesis. Okay, and I was able to make sense out of how it all fits together and see the whole picture. No one else does that. No one else writes about the whole picture. Each one is sort of like on their own little silo of what they do. So when it comes to neurodegeneration, I'm the great unifier who figured it all out and put it all together. The other great theory in neurodegeneration is the Jack Delatorre theory, and I call that the mouse equivalent theory. I spoke about that earlier. And there's a lot of other brain toxins, and most of these are mitochondria inhibitors, okay? And I made sense out of all that stuff, how they're excitotoxins, because you can't make the ATP to pump out the calcium, okay? And there's tons of them. Aluminum, it's in your tap water, it's in the trails in the sky, the you know what? Trails, okay? Lead, okay? That's a contaminant, not that common nowadays, but it still happens often enough, and it was a big problem for the ancient Romans. Hg, mercury in high fructose corn syrup. It's a mitochondria inhibitor. Cadmium, it's a mitochondria inhibitor. It's like I won't, like brake pads will give that off. That's why I won't eat at a sidewalk cafe in a busy street, because you're going to have increased cadmium inhalation. Antipsychotic medications, antidepressant medications, anti-seizure medications, all mitochondrial inhibitors. So they're all going to worsen excitotoxicity. Fungal inhibitors, typical thing in processed food, so it doesn't spoil, it prevents mold growing on their antifungals. Very often, mitochondria inhibitors. So a lot of these antibiotics, aminoglycosides, fluoroquinolones, a very commonly used antibiotic. Fluoride increases transmission across blood-brain barrier, blood-testicle barrier. So these are very commonly used for sexually transmitted diseases, for brain infections, and they get across the blood-brain barrier, and they can be toxic to the brain. Okay, TCE, trichloroethylene, that can increase your risk of Parkinson's disease, is also a mitochondria inhibitor. Hydroxynonenal from omega-6 cooking oils from fried foods, mitochondrial inhibitor, and it also predisposes to lipid peroxidation, which can damage membranes, including plasma membranes and neurons. Okay, excessive dietary fat, especially sat fat, but all fats eventually can lead to mitochondrial inhibition and diabetes-related insulin resistance. Okay, let's see, anything that leads to abnormal neuron cytoplasm calcium is going to increase the risk of neuronal death and apoptosis. And you see there's a tremendous number of things all converging on elevating cytoplasm calcium, leading to increased metabolic rates, and there's a lot of things dropping that oxygen glucose delivery and ATP production. So these neurons are screwed. And that's why you see all these pathetic patients. Like I said, my internal medicine friends, they'll tell me, every single one of their 60 and over patients is cognitively slow. When I say cognitively slow, that means things like you talk to them, they don't have any zip vitality, they don't have any quick brain processing. They tend to have preserved social skills. Like a typical old person I'll talk to is hi, yes, thank you. You know, they're nice, but they're real slow mentally. And that's why doctors always talk in sort of a polite, dopey way. Hello, Mrs. Jones, nice to meet you. Because until proven otherwise, they're used to talking to cognitively slow people all day long. Nice thing about vegan diet, you got good blood supply. You don't plug up your arteries. The main reason people get stupid, they got all these things going on with excitotoxin effects, and they plugged up all their arteries from chronic atherosclerosis. I look at their brains, their necks, and their chests on CAT scans, and I see more often than not all these arteries are heavily calcified. You calcify all your arteries, that means the arterial wall is dying, okay? That's an indicator of diffuse damage to the arterial system. Okay, that's another reason why, too, a lot of people always say, well, I'm cutting down on oil. I'm cutting down on processed food. And I'm like, look, you dummy, you don't understand. You're already, behind the eight ball, half screwed. So fix yourself as fast as you can and be 100% good so you'll have a chance to keep your arteries open and your brain functioning. Because, once a person's stupid, their mental curiosity just gets worse and worse. And the likelihood of them ever fixing their own problems becomes essentially zero. Okay. This was just a mnemonic I made for my theory of dementia. And so I was boasting a little bit. So I made the mnemonic boast. Brain function depends on OGD, oxygen glucose delivery. And I went through all the things here that'll diminish it. Having antioxidants protects the brain. Well, where do you get them from? You get them from plants. How do you open up your arteries? You get all the good stuff from plants. The vasodilators, potassium, P for plants, P for potassium. Magnesium's in the center of chlorophyll. You get that from plants, okay? Sodium is mostly in processed food, and they add it to meat as both a preservative and a flavorant. So that's the bad stuff. So the bad stuff tends to all come from meat, oils, and processed food. The good stuff comes from whole plant foods. Let's see, what else? Stimulants. These are all things that increase the metabolic rate, making it harder for the oxygen and glucose delivery to reach it. So all these stimulant things, we talked about that. Toxins, and we talked about all this stuff. Mitochondria inhibitors, circuit inhibitors, heavy metals, mitochondria. A lot of these things are all mitochondria inhibitors. A lot of these endocrine disrupting chemicals are actually also neurologic disrupting chemicals like BPA. Bisphenol A, that inhibits mitochondria, okay? In the next lecture, I'll go through this mitochondria inhibition in more detail. But the key point to realize from this lecture is neurovascular uncoupling. When your metabolic rate is disproportionately increased relative to your oxygen and glucose delivery, eventually that nerve goes into apoptosis. And that's why I think, whenever I hear somebody claiming they know about, helping the brain and they think caffeine's a good idea, I lose respect for them, because how could caffeine be good for your brain? Number one, it impairs your sleep, and your brain needs sleep to clean itself for the glymphatic system, okay? And then number two, it drops brain blood supply while simultaneously increasing metabolic rate, meaning it increases your risk of apoptosis. How could that be good? And then the same person who says caffeine and coffee and tea are good for you is going to tell you, oh, stress is bad for you. Yeah, right. Don't you understand? You're talking about the same thing, genius. Okay, the word in Polish for genius is geniusz. So when everybody does something stupid, a Polish person will say something like, good job, geniusz. Sarcasm, genius, all right? So that's stupid. All right, let's see, what else? And, okay, well, I think I kind of made that point. All right, and so I was boasting in the sense that I'm bragging that I think it's funny. That's why, to me, when somebody says something like, oh, that's what they say in the Ivy League or Harvard or something. Look, I did part of my training at Harvard. I did part of my training at Stanford. And I know what a joke those places are for this sort of stuff. Almost all doctors in conventional medicine will just go with the party line, whatever it is, no matter how stupid it is. You're trained as a doctor to memorize stuff and follow the standard of care. You are not trained to think. You are not trained to ever challenge the established ways of doing things. There is no reward in medicine for challenging the established ways of doing things. But, like I said, I've told you earlier in this book what a joke Alzheimer disease is, okay? They can't diagnose it and they can't treat it. The whole thing's a big joke. And I've explained to you how you can make sense out of what to do to protect your brain cells. And it's rather obvious. So, and I've also been pissed off because I'll try to publish these articles in conventional journals. They won't publish them. If you're promoting a drug or showing how the benefits of a drug, you'll be published automatically. They'll call you on the phone. They'll set up interviews for you. You explain something that doesn't help their drug world, they don't want to talk to you. And, this video will probably get shadow you-know-what bond because it talks about all these toxins and it criticizes all these commercial foods. But that's my job, to be the bad boy of veganism. Okay, so the next chapter will be mitochondria inhibitors. I hope this was helpful. You got the neurovascular uncoupling theory of Peter Rogers, and you also learn a little bit more just in general about calcium metabolism and excitotoxicity. So hope it was helpful to you.